Good evening, everybody. Hello and welcome. What a fantastic crowd. Hello, friends. Welcome on behalf of all my colleagues here, our team of staff and students at the Botanic Garden. I want to welcome you to the opening lecture of the Spring 2025 Bulb Show. I'm John Berryhill. I'm the director of the Botanic Garden here. And before I invite Marge on to introduce Professor Jess Gersony, I just want to take a few minutes to share some important news and some well-deserved thank yous. First, as always, after the talk, I hope you'll all take a walk with us down to the Lyman Plant House, which will be lit up, and get a sneak peek of the Spring Bulb Show. The easiest way to do that is out of these doors here. If you came in that way, you can also sneak out the doors to my right, your left, and then turn an immediate right. We'll just spit you out right by Landing Fountain. It's the easiest way to get there. I should have said happy spring, too. The witch hazel right behind me is already in bloom and fragrance, so if you're walking around in the weeks ahead, just give that a sniff. You'll be glad you did. But down at the Plant House, not just a chance to see the flower show. I'm happy to say we have several exhibits there going at the moment. Of course, the Eunice Rothman piece, Haba, that's part of the collaborative exhibit here now in collaboration with the Smith College Museum of Art and Seeds. Will be there, as well as some really exciting new exhibits by students, including Botanical Bodies, which is a really amazing new exhibit that went up, put together, conceived, and created by our amazing Botanic Garden student educators. And there will also be two student art installations by Ella Wang and Jamie Biagirelli that will be part of this year's Bulb Show that are really stunning and have been worked on for over a month now with the help of our uber-talented greenhouse horticulturist, Lily Carone. Yeah. And of course, none of this would be possible without the incredible work of the whole Lyman horticultural team. Again, Lily Carone, Jimmy Grogan, Dan Babineau, Chris Chae, Sophia Holmes, and especially senior Allie Wernell, who again this spring, we're so grateful, Allie, for being the lead designer on this year's Bulb Show, as you were in the fall. The work you've done for us is amazing, and we're tremendously grateful. I also want to thank those who did so much work to plan and coordinate and help prepare for tonight's event, and for the 2 weeks that lie ahead, including Sarah Loomis, Julie Thompson, Sherry Peabody, Liz Whiteley, Gabby Immerman, Angela D'Souza, Ben Green, Andrew Robello, Nate Sachs, Dave Dion, Elio Berkovic, Sophie Picard, Charlotte Muse, Ella Sommerfeld, Sophia Zucala, Abigail Dlug, Molly Gottfried, Riza Baldonado, George Kerman, and of course, the many amazing volunteers who do so much work for us. They're all the welcoming faces that guests see when they come and visit the Botanic Garden. Yeah. Thank you for beating me to it. I was going to say, so many of those people are in the audience today, and can't say enough about the work you've done and how grateful we all are. This year marks a new chapter for the Botanic Garden as we move forward without the Fall Mum Show. This change is not about doing less, it's about doing more, and I'm thrilled to announce that our first new botanical exhibit will be coming this May. It will offer a behind-the-scenes look at some of the hidden stories, facts, and numbers that shape our greenhouse collections and Lyman itself. As someone who's spent many years here, I'm excited to discover even more about this beloved place, and I promise that all of you that come to see these exhibits, it will invite you to see Lyman and our collections with new eyes. Lastly, to all of you who support us as Botanic Garden members, thank you so much. One of the most striking lessons I've learned as director is how vital that support truly is. We simply could not do what we do without you. If you're not a member, I encourage you to join this community. Membership comes with some really great benefits, like free admission to many Botanic Gardens around the country. It's a great way to earn your donation back very quickly. And it's a wonderful way to support us as you explore more green spaces. It's really an honor to have Professor Gersony with us tonight, and I'm going to, she's been a dear friend to our whole community here at the Botanic Garden, and as we at the Botanic Garden have learned a great deal from your approach to your work And while I'll leave that full introduction to Marge in just a moment, I want to take a moment to reflect on the Botanic Garden's place in this community. We recognize that our work takes place on the unceded land of the Pecumtuck people and the Nonotuck people. And as we just strive to serve a vibrant and diverse community of students, we know that there's still so much learning and listening to do. Addressing these challenges calls for bold thinking, the kind of thinking that we look forward to hearing about tonight. I'm here not just as a host, but also as an eager learner alongside of a community committed to growth and meaningful change. Now, it's a joy to be able to introduce a very dear friend to us, Marge Poma Alarcon. Marge is a recent Smith graduate and a multi-year veteran with the Botanic Garden on our student team. She began with us through work study during her first year here, where I was lucky enough to work with her every single week. Still cherish that time. Marge quickly connected not only to the physical work we do, but also to the complex, meaningful conversations that shape our mission, especially our efforts to create an inclusive learning community. Her work with us continued as an intern and as a vital partner in connecting the Botanic Garden with the broader Smith community. Today, Marge is a graduate in Dr. Gersony's Place Lab, studying the effects of drought on northeastern trees. And working with Marge, I have the privilege of working with Marge to facilitate weekly gatherings for her fellow plant-loving Smithies to explore botanical topics every week, and I'm very grateful for that. Please join me in welcoming Marge Poma Alarcon.
Thank you so much, John. I have the immense honor of introducing tonight's speaker, Professor Jess Gersony. She is currently an assistant professor of biological sciences, having taught courses such as plant physiology and understanding climate change through plant biology and the arts. And she is also an instructor for an intermediate tap dance course here at Smith College in the dance department. She received her PhD in organismic and evolutionary biology from Harvard University, where she studied carbon movement in plants and worked as a postdoctoral fellow at the University of New Hampshire, where she developed partnerships with local ecologists on scaling the physiology of leaves to the ecosystem level. She is continuing this work here at Smith, and it is generously funded by Smith College as well as the National Science Foundation and the U.S. Department of Agriculture. By day, she is a plant ecophysiologist who studies plant stress with regards to climate change, which has taken her from the tundra of the Arctic to the deserts of Chile. By night, she is a professional tap dancer and poet, having performed in multiple cities and published in the Sycamore Review, respectively. But her proudest accomplishment thus far, as she exclaims, is the foundation of the Plant Physiology, Arts, and Community Engagement Lab, or as we like to call it, the Place Lab, here at Smith. So tonight, I also have the honor to speak on behalf of the Place Lab, which is composed of 3 pillars: the science of plant physiology, how plants transport water and sugar, and what might happen when they lack these essential elements; the creation of art, which allows us to comprehend the complex structures necessary for plant functions and communicate our work to a broader audience; and community engagement, which exists within our department and beyond the campus to encourage shared resources and collaboration with our local communities. The culmination of these 3 core principles fosters a feminist, anti-racist, and social justice and equity-centered lab experience, pushing back on white supremacist values that often turn STEM research into an inaccessible field of study. This means that every day, the Place Lab is filled with pure joy, hard work, and celebration as we work together with plants and people for the love of seeing our planet, our home, thrive in our collective future. All of which couldn't have happened without the amazing members of our lab, and of course, Jess. Please join me in welcoming Professor Jess Gersony.
Hello, everyone. Oh, let me choose the thing. Can you hear me? Does that sound good? Okay. All right. Hi, everyone. Thank you so much, John and Marge, for this introduction. Today, I'm going to be talking to you about climate change, plants, and longing. But more specifically, we're also going to have an artistic and scientific exploration by a reimagined research laboratory. But before I get into any of that, I want to make sure I ground this talk in appreciation and love, because that's the vibe. So I just want to take this moment to talk about this. I feel so lucky to get to do this work that I'm going to talk to you about today with the amazing people in my lab and our collaborators. This job and the people I get to work with feel like a dream come true. I don't know that I've ever had such a kind, warm, joyful, and honest group of people who I get to call my team. Throughout the talk, you will see many pictures of them, but specifically, I want to take a moment to thank Marge, without who the Place Lab would not be possible. She is such a thoughtful, kind, curious, and brilliant thought partner since day one of me coming to Smith. I also would like to thank everyone at the Botanic Garden for giving me this opportunity and this beautiful platform to speak tonight. Specifically, Lily, Sarah, John, Gabby, Julie, Jimmy, everyone. I would also like to thank Smith and the bio department for supporting this work in so many ways. And lastly, I also want to thank my wife, Stage, who is here tonight, Dr. Moon, who's my high school science teacher, and my family and friends who lift me up and give me the strength to try and move through this world in a reimagined way. And I also want to thank all of you for coming tonight. What a gift this is. Okay. So let's get into it. Okay, so I'm going to talk about two different things. First, I'm going to talk about our broader project of reimagining in the Place Lab, and then we're going to have a little mini vignette story about a scientific and artistic exploration of plants and climate change. So, regarding the broader project of reimagining, in order to put that in context, I want to take a step back and talk about the state of plant sciences in regards to DEI principles. So, the field of plant sciences and STEM fields generally contain vast systemic issues and inequalities. And one key systemic issue is the underrepresentation and historical exclusion of certain groups. And this is current. This is all still really in real time happening. We see underrepresentation based on race and ethnicity, on gender, on disability, and these are all papers that have come out in the past few years. This is not old data. This is current data for the field of plant sciences. And it's really important to note that this is due to a history of exclusionary practices and systems, including colonialism, sexism, ableism, white supremacy, and more. And I'm not going to go into too much depth for each of these because we only have so much time, but this was a great paper that was looking at the impact of colonialism on ecology, and the way that they showed it was showing this map that shows the number of bird species named after European surnames, and you can see the vast majority of them are not in Europe. There's also still a lot of sexism and ableism in our field, specifically thinking about STEM. So this shows that female interns with ability issues are viewed as having lower field aptitude than male interns with ability issues. And they generally show how sexist beliefs, workplace issues, and intern gender can affect responses and assessments by potential mentors. There's also LGBTQ discrimination still happening. Again, like this is 2021. So they saw that LGBTQ STEM professionals were more likely to experience career limitation, harassment, and professional devaluation than their non-LGBTQ peers. There's also some more ableism happening. This was another paper that came out that showed much of the research framed disabled students as a problem to be solved and focused only superficially on student sense-making. And lastly, but certainly not leastly, we have white supremacy culture. So this was a paper that came out a couple of years ago, and it's a really, really beautiful paper. And all these papers I'm citing at the end of the talk, I learned how to make a QR code so you can scan it, and you can access them if you're interested in reading more. But so this was a great paper that was thinking about how is white supremacy culture manifesting itself in STEM fields and science communication fields. And what they did, I'm obsessed with how big the screen is. This is awesome. It's so cool. Okay, focus. So in terms of white supremacy culture, it's good. We should bring some lightness. So there's an amazing paper called by Jones and Akun in 2001 that names different pillars and characteristics of white supremacy culture. So things like defensiveness, either-or thinking, fear of open conflict, individualism, objectivity, only one right way, and paternalism. It goes on, perfectionism, that's a big one we think about in the lab, power hoarding, quantity over quality, right to comfort, sense of urgency, and worship of the written word. And so, yeah, like I said, they go through defining the trait, they're defining the characteristic, and then also talking about how it manifests. And just to pick two out to just get a little bit deeper on them, so this one's quantity over quality. And this manifests in, it causes scientists to focus on their own agendas and needs rather than the audience's issues and needs. And it prioritizes research and publication, devaluing public outreach or community-based projects, and it feeds the stereotype that scientists are cold, impersonal, and distant. Another one, sense of urgency. This causes scientists to sacrifice relationship building in favor of action, including relationships between researchers and diverse communities and stakeholders. Okay. So this is heavy, right? This is the context in which we're existing as scientists. But thankfully, there are many people, primarily BIPOC scholars, who've thought about how to move forward in a reimagined way. So this is just 5. There's a bunch of these papers out there written by BIPOC scholars about new ways forward and different actions you can take. And I'm just going to show, luckily each of them, they have awesome figures, so I'll show you some of them. This one is a paper called Recreating Wakanda by Promoting Black Excellence in Ecology and Evolution. And one of the things they suggest is requiring Black history courses and teaching research through a justice lens. This is another one about an anti-racism plan for a geoscience organization, and just picking someone that we are kind of engaging with in the Place Lab actively, investing in recruiting from minority-serving institutions, and collaborating with communities as stakeholders. This is another one about ecology and evolutionary biology, uplifting BIPOC scholars, and this one they break into different levels of the system. So you have supervisors, departments, or institutions, and things that you can do at those levels. Again, it's amazing. These papers are amazing. This one is thinking about learning from and amplifying the voices of BIPOC scholars and incorporating different perspectives within EEB research. Another one about anti-racist interventions, thinking things like implementing healthy expectations for work-life balance and acknowledging racist histories and barriers. And the last one I'm going to show is a paper that's more focused on decoloniality. And they talk a lot about this concept of decolonizing and shifting our mind and our expertises in the context of STEM. And this one is where art comes in. So they mention specifically the use of art to help increase communication on interlinked biological and cultural diversity, and then collaborations between artists and scientists and between indigenous people and colonizers can result in even more creative ways to exchange and mutually enrich knowledge and understanding through innovative art science projects. And so that's really what we're going to get into for the second part of the talk. So with all of this, motivated by this context, I started the Place Lab here at Smith, Plant Physiology, Art, and Community Engagement. It's motivated by all these papers that I just talked about, but also the broad readings and literature in the broader fields of translational ecology, STEAM, indigenous studies, and anti-racism and Black liberation futurism studies. And through this, we really try and center community building, art, and justice. I'd be completely remiss if I did not mention the Great Queer Lab. This is a really, really radical and inspiring lab that is based in northern Canada and run by an Indigenous and queer researcher named Max Liberon. And the Clear Lab is a feminist anti-colonial laboratory, which means our methods foreground values of humility, equity, and good land relations. And we really use this lab as kind of our dream, like our model for how we're trying to do this work. Okay, so we started the Place Lab. This is drawn by Marge. There. Okay, so the Place Lab stands for Plant Physiology, Art, and Community Engagement. What is it? Most importantly, it is the people. These are the amazing current members of the Place Lab. We have Bella, Flora, Avery, Al, and Mia and Chloe, who are current undergraduates. And we have Malini, who's our postdoctoral researcher, you know Marge. We also have Teresa, who's our PhD student, Maxine, who's our research technician, and Iz, who is an alum, who's still working on a few projects in the lab. Could you all please stand up so we can all applaud them? Woo! Thank you. Yeah, okay. I'm gonna move on, but okay. So thinking about our lab culture, I wanted to present some of the pillars of how we define and work through our lab. The first one, and I think the foremost one, is this concept of gentleness in STEM. So I went to an Academics for Black Lives conference a few years ago, and I heard this phrase, and my mind exploded. I'd never heard the words gentleness in STEM in the same sentence in my entire life, and I'd been in STEM. So I was really blown away, and I really use this concept whenever I'm faced with a problem in the classroom or in the lab. I'm like, what is the gentle way to move through this? And my lab very much embodies this. We also work to bring our whole selves into STEM spaces. We always check in with each other at the beginning of meetings, just on our humanity before we get into the science. We center care. This is a line from the Clear Lab. If you are brokenhearted or sick, get out of here. We want you to take care of yourself, and we'll figure out the job later. We also work to bring art into STEM spaces, which we'll talk a lot about. We have an apology protocol. This work is messy, and we mess up sometimes. And we have a protocol, which is in one of the zines, which I'll get to in a bit. And we always make space for humor and joy. That's very much the heart of the lab. And I should say, we're working on codifying this into something called the Place Lab book that we're working on. And you can read more about in the zine, but I'll talk a bit more about that later. So we have a lot of projects going on in the lab right now. I am not going to talk about all of them. In fact, I'm just going to talk about one of them. But I'll just give you a sentence for each of them. If you want to ask any more questions, I'd be more than happy to talk about it, or any of the lab members would also be happy to talk about them. Okay. All right, so we try and have justice-centered pedagogy. So in the classroom, in my plant physiology class, in my upper-level classes, I always try and frame the work in context of justice, so thinking about environmental justice issues as the lens through which we're learning physiology. So thinking about issues of like urban tree inequities, or thinking about climate justice or climate change as a justice issue, or thinking about sustainable agriculture. Those are kind of the lens through which we learn about photosynthesis and respiration and xylem and phloem, which we'll talk all about. We also work on co-producing research questions with various communities invested in the forest. So stepping a bit outside of academia and talking to the people who are invested in the forest in all different types of ways. So we do that through some work in Holyoke with my dear friend here, Yoni Glogauer, who's the Director of Conservation and Sustainability for the city of Holyoke. We also talk to different forest managers and people at the U.S. Forest Service, and think about how to implement our research into their models and different programs and platforms that are used by foresters on the ground to try and optimize their forestry practices for climate solutions. We also try and center diverse perspectives. We got a grant from the Interdisciplinary Making Initiative here at Smith to bring in Andre Strongbear-Hart-Gaines, who's an indigenous Nipmuc artist and activist, who came and lectured to the class and also led a community event. That was really amazing. We also have taken trips to Soulfire Farm, which is an Afro-Indigenous farm in northern New York, and got to learn about their really robust and inspiring reimagination project. We are also collaborating with Girls Inc. in Holyoke in terms of thinking about educational justice projects. And then lastly, we're doing some art and science integration, and that's what the rest of the talk is about. Okay, but if you're interested more in the reimagining projects, so Marge, Teresa, and Iz they're really large. Okay. They're working on a manuscript about this work, so stay tuned. Hopefully it'll be out in the next year. Okay, so that was about the broader project of reimagining. Now we're going to move into a scientific and artistic exploration of plants and climate change. Settle in. Okay, so why science and art? This is one of my favorite lines from one of my favorite poems by one of my favorite poets. This is a poem by A.R. Ammons called Corson's Inlet. And it's the quote that I allow myself eddies of meaning. And this is really at the core of what I think about when I think about understanding and knowledge. What is an eddy? An eddy is one of these circular movements of water. You can see them here. They're so beautiful. Marine for Paulette. Okay. So thinking about eddies of meaning. And I really fundamentally believe that we can't understand climate change until we understand the Earth. And we can't understand the Earth through one discipline alone. I don't think looking at scientific papers or doing scientific research projects will give you the full, robust view. I mean, I think it takes all different types of perspectives. And this is why we integrate across different ways of knowing. So what do we think about in the lab? We think a lot about carbon and water movement in trees during environmental stress. And for this presentation, I'm going to present to you eddies of artistic and scientific exploration around these concepts. And so to broaden, or actually to shift the framing a bit, another question we really think about in the lab is, what does it mean to hold on to water as an ecosystem, as a plant, and as a person? And even broader, you can step one more lens back and say, what does it mean to hold on to something being lost? And this is really the heart of our work in the Place Lab right now. And so I'm going to present 5 different eddies about this concept, and you'll see how they're kind of all intermingling with one another, hopefully. So the first one is about climate change and dragging rivers. So you might have seen figures like this before, that anthropogenic climate change is really, really happening. This is a figure from the Intergovernmental Panel on Climate Change. And what you're looking at is the average change in surface temperature from the beginning of this century to the end of this century. And this is in a really good scenario, like if we do everything possible to combat climate change. And this is a worst case scenario if we really don't do anything. And you can see we're getting into very, very high degrees. And this is the same down here, but for precipitation, and the lighter color is drought. And so I present a lot of figures like this in my classes, in my lab. We're always talking about them. We're always looking at them. And it's a lot to take in. And it's that question of taking in, like what does it mean to actually internalize this data and reflect on it? So that's where the art comes in. And so we make a lot of art in our lab to really try and embody and understand this work more than just through numbers. So during this presentation, I'm going to show a few different pieces of art, and I've been advised to take a beat. So I'm going to put the art up there, and then I'm not going to say anything for like 5 seconds so you can process it, and then I will start yagging again. Okay, so here's the first piece in response to thinking about climate change. All right, this is a personal meditative piece about climate change by Flora Meeker, exploring a changing landscape due to climate change, close to where they grew up. And to quote the artist in thinking about the piece, "The Cranberry Glades Botanical Area is one of the landscapes that makes up who I am, both my identity and my physical body shaped by our overlap. When I say mending a broken heart, I mean turning to embrace this grief of change so far outside my control, knowing that I can't ignore the daily grief, but that my only antidote to grief is love's tending. It's a question. How do we honor our grief while committing ourselves to adaptation in the face of a precarious future? And how much of yourself do you lose when you turn away from the pain of how together we are changing?" And so I'm going to take a moment to read the poem in the bottom right. Mending a Broken Heart. "The planet has changed utterly. As you read this, your heart is pumping. Electric sparks, flowers and stems, rhythms and echoes. Count. Stop to look and listen. Presence and absence, narrowness and refusal. Go on taking care of them. This seal keeps you." So this is the piece Precarity by Flora Meeker. And now I'm going to share another one by Avery Maltz. This is a piece by Avery, who developed a method for printing photographs on wood and then preserving them using natural plant-based resins. It is a photo of the Dear Dawn redwood on campus. And to quote the artist, "This whole semester, I've been processing the heavy feelings around the reality of climate change, as well as my own personal feelings of loss, and each art piece that I've made has reflected that process. In thinking about tree death, climate change-induced death, and the ephemerality of everything that we know and love, I'm comforted by the beauty of the present moment. It feels miraculous that we get to share this blip in time with trees, however fraught the current moment may be. There is such joy in the conscious witnessing of nature, and because it is a dynamic living system, that joy is coupled with the grief of transformation. We are all ephemeral living systems. I hope to create a snapshot, a portrait, to turn a mirror on this deep river of joy and grief that is always running beneath us." So these students, they're amazing. All right, this piece is in response to tree mortality, this piece is in response to tree mortality in regards to drought. And drought is occurring here in the Northeast. So here are 4 different images from 2016, 2020, 2022, and 2024 by the Drought Monitor, which is a USDA product. It's awesome. You can well, it's awesome if you really like learning about drought, but it's a very cool website, would recommend. And so what you're seeing is, yeah, severe droughts are occurring. If you live here, you kind of know this has been happening almost every other year for the past few years, and it's predicted to get worse. The northeastern US is also experiencing exceptional warming. So this is a paper from a couple years ago showing the differences in temperature between 1902 and 2018. And you can see that Connecticut and Massachusetts are in fact warming faster than anywhere else east of the Mississippi. And that's due to things like ocean currents and winds, but also, of course, anthropogenic climate change. I actually got this figure from Professor Jesse Bellemare because he presented it, and I was like, that is the best way to visualize this. Very depressing, but it's important to have good visuals for these things. Okay, so this is from the Union of Concerned Scientists document, and what you're seeing is the temperature and the heat index for Massachusetts in different emission scenarios. So the yellow is best case scenario. So best case scenario, we have the climate of around Maryland by 2070. Worst case scenario, we have the climate of the Carolinas by 2070. So this is real, and it's happening, and it's predicted to get worse. And so when we think about climate change, we're thinking a lot about movement and changes. We're thinking about shifting water from one area to another. And that's a lot, it's kind of gotten into my psyche, and so I want to share a poem I wrote related to this. Ancient Tillites. Yes, I believe in geology, the igneous, the sedimentary, and the metamorphic. You sit at the kitchen table, exhaling. At least try to fill the space of your container. My head tilts to its shoulder with weight. Outside, a boulder, a glacial deposit. I drag my feet to the river, and I drag my river home. So we're thinking a lot about water movement, right? We're thinking about it in the context of drought and climate change. But it's, of course, also happening in plants. We have water moving, gushing through plants all the time, and so I would be remiss if I didn't show you some sick images of the inside of plants. Okay, well, I kind of just gave it away, but I was going to ask you to think about what do you think you're looking at. But I'll take a bit. What do we think we're looking at? Just take a second to reflect. It's so cool. Okay, I'm clearly not good at waiting. This is the inside of a leaf. And so if you took a leaf and you cut it, and you're looking at that part, so this is the top skin of the leaf. This is the bottom skin, also known as epidermal cells. And then these are mesophyll cells. My plant physiology class knows. Yeah. All right. These are palisade mesophyll. These are spongy mesophyll. That's where all your photosynthesis is most of your photosynthesis is happening. You also have things like veins. Veins are the things you see on the leaves, but they're also inside of the leaves. And they're tubes that go through the whole plant, all the way from the roots, connected all the way to the top of the leaves. And this is a scanning electron microscope image. And here's another one. It's a little bit fuzzier, but you can actually see more. So if you look here, you can see kind of jelly beans inside of the big jelly bean. Those are chloroplasts. So that's where photosynthesis is actually happening in the thylakoid membrane and the stroma. But you also can see a stomata. Stomata are very special cells. They're arguably the cutest cell of all, and I'll show you some more photos. So this is the stomata. Oh, they're tiny, tiny little mouths that exist on the leaves that allow air to come in and come out. And shout out xylem, too. Xylem's awesome. Xylem are the pipes in the plant, so the actual pipes that are moving water from the soil up to the atmosphere through the plant. And a lot of people, including myself, I'm always thinking, how big are these things? It's a very good question. It's about a hair. The xylem is about the size of a single hair. So look at that. Appreciate it. Okay, so we have hundreds and hundreds of xylem in your plant, and that's allowing water to move. And so I'm talking about this water movement. And this is one of my favorite questions and facts I say in every talk I ever give, which is the question, how much water do you think is... So you have a red oak tree, beautiful, my favorite. Red oak tree in the forest, and it's doing its thing. It's a happy, healthy tree. The question is how much water is moving from the soil to the leaves to the atmosphere in one single day in terms of gallons. And I usually get an answer around like 5 gallons, which is a lot if you think about those 5-gallon jugs. But it's actually 100 gallons 100 gallons are moving through one oak tree in one day in one forest. So these tiny, cute little cells are actually regulating our global water system in like a very acute way. So it's really, they're impressive. I could go on, but I won't, for the sake of time. Okay, so, well, I'll go on a little bit longer. So here, just because, look it up. Okay, so this is a figure I use a lot in my classes to kind of think about water and carbon movement through the plant. And so just to track the journey of a water molecule. So the water, we water our plants, right? We water it into the soil. It gets into the plant. It goes up the tube, the xylem, the hair. It goes up the tube, and then it goes out of the stomata. And the reason why it's actually coming out is because the stomata is opening to let CO2 in. So the stomata is opening, CO2 is coming out, and water's like, get me out of here, and it like rushes out because the air is drier than the inside of the plant. We can talk more about that anytime. But so what we have is CO2 is then coming in, photosynthesis is happening in those jelly beans I showed you, and then it's getting turned into sugar. The sugar is being loaded in something called the phloem, which we'll talk more about because I love it, and that's what we study. And so we have sugar moving into the phloem, that's drawing in water, and then that's being pushed to areas in need of sugar. And so this is kind of how the plant works. And so in terms of water and carbon. And so we think a lot about in the lab about this gushing of water, right? This 100 gallons that's moving through a tree every day. And Marge, you all know Marge. So Marge came up to me my first semester here, and we were making art projects, and Marge was like, Jess, we're talking a lot about this is me paraphrasing, we're talking a lot about this water that's coming out of the leaves. Like, I want to collect it, and I want to use it to watercolor a painting of a tree, like to really kind of embody the whole process. And I'm like, oh my God, unbelievable idea. That's impossible. I was like, that's way too hard. Stomata, despite their cuteness, are very finicky. It's hard to know exactly when they're open or closed. But so I was like, Marge, you got 2 weeks. I tried cool, but try something easier. And then, of course, she did it. She spent 2 weeks learning about the patterns of stomata and like putting a bag on a leaf and running out when the sun was out, taking it back and getting all the water. And then she watercolored this beautiful painting of a tree outside where she grew up in New York City. And it was specifically in the context of thinking about urban heat island effect, hence the raising temperatures and the burning. Oh! Obsessed with that response. Yes, that's correct. Great. Okay. Okay. So we're thinking a lot about flows in plants, right? We're talking about water gushing everywhere. Well, in the form, it's in humidity. It's not really like a tidal wave. But anyway, we're thinking a lot about water loss. But if we think about plants need water in their cells to do everything they need to do. So the question our lab is always asking is, how does the plant hold on to this water? What is it actually, what are its talents, what are its skills? Are different plants doing it differently? What does it look like? And so we think a lot about this concept of holding on to these waves of water. So specifically, the question we're asking is, when water is being lost, so because of climate change or drought, how can the cells inside of the plant hold on to it enough to keep the plant alive? And this is like the heart of the research questions I'm going to present. And so one of the main tools is osmosis. So osmosis is the carrying of water in my understanding of it. But generally, we can think about it that the phloem and photosynthetic cells need water in order to function and keep from wilting, right? We've all seen it. They wilt. It's really upsetting. But it's upsetting not only visually, but also physiologically. The plants are really not happy when they're wilted. They can't do photosynthesis. They can't do some of their key processes. And so they need water. And the more sugars and salts inside of the cells, this is key, inside of the cells, the greater their ability to hold on to water and maintain functioning during drought. So we really want to pack leaves and the phloem with sugars and salts. And so we ask a lot of questions, like how many sugars and salts do different species have, and how do they vary, and how do different cells vary? And this is the heart of the work that I've been doing for the past 10 years. And now I'll share with you 2 more poems I wrote about this. All right, this one's called Introduction to Hydroscapes. It was the day you tried to hold on to individual ocean waves. It was the day after a satellite was probably launched or volcano. An animal was born. Did you ever think to not think harder? I thought to erase water. But the animals, any animals, the animals didn't believe any of this. Now I'll share one more called I Could Collect a Lake from my chapbook called I Could Collect a Lake, published by Bottle Cap Press. Okay. All light of yesterday in this place is now hidden. It could be the ground heating my feet, or it could have fled with the stream into some larger body. I wait for the moon, but for the trees, the moon means only tides pulling something closer. Could all water have been in this lake? I could collect a lake. I am a lake, 1,000 feet deep, deep blue with white caps trembling. The moon pulls and I am flattened, hollowed, and stretched thin. Okay. You're very sweet. All right, so we think a lot about this question of holding on. What does it mean to hold on to water, and how do we do it? And so we try and shed some new sight onto these questions. And so I'm going to take a few more minutes to talk about some of our recent research findings. So one question we have is, how does the phloem hold on to water, right? All this water gushing out, we have climate change happening, we have more droughts. What is the phloem doing? And we don't know a lot of what the phloem's doing because it's really small. So I talked about the xylem is like this. The phloem is a small fraction of that. So it's even smaller. It's also alive, and it's really finicky. So if you touch it, it stops moving. And if you puncture it, it clogs. And it's a nightmare. It's great for the plant. It's harder for the scientists. And so the main method in 2025 for studying this is amazing, and I'm going to share it with you. So scientists many decades ago, 4 or 5 decades ago, were like, bugs do it. We have all these bugs that are poking their little mouth parts, and then they stick it into the phloem and start drinking the sap. They're called sap suckers, some of them. And so they're doing it, so let's take advantage of them. And then so scientists were like, let's cultivate a group of aphids, bring them to our study tree, let them feed, let them mess around, find the phloem, and then they laser off the body of the bug, leaving the mouth part so the phloem sap can come out of it. And this current method, 2025, this is the bug, it's feeding, you laser off the body, you get the little mouth part, and then you have this tiny little thing of phloem sap, and you bring the phloem sap back to the laboratory and you study it. As you can imagine, very hard, a very hard method. And so therefore, we really don't know a ton about the phloem. And so I spent a few years during my PhD being like, we gotta have another way. I can't be doing that. And so I collaborated with this amazing scientist, Dr. Arthur McLeland, here, thinking very deeply about our predicament with the phloem. And this is his magical tool called a Raman spectroscope, or Raman spectroscope. These are awesome. These are the future. The way it works, well, let's be clear, I don't know exactly how it works. That's deep physics. But the way I understand that it works, so it shoots lasers at whatever you're interested in, and then it tells you the chemical composition of what you're looking at. This is magic, in my opinion. And they do it in all different fields. It's used in dentistry and geology. It's even used in art history. They shoot the laser at old pieces of art to figure out the chemical composition of the paint and of the pigment that was used centuries ago. So cool. That's it. Shout out the ramen. You put your little leaf here. So anyway, we spent 2 years trying to figure out how to use this tool to look at the phloem, and we eventually found out that if you cut it in a specific way and you bring it back to the lab and you know where the phloem is, you can shoot the laser at it, and it'll tell you the amount of sugar in the phloem, essentially. And so the goal of this project was to see sugar on the sugar highway. And so here are some of the results, but all we care about is this little green line, and that is the predicted sucrose concentration, or the amount of sugars in the phloem. And what we found was it's very sugary. After 2 years, that's the answer. So it's around 1,500 millimolars of sugar in the phloem. And there were studies from literally a century before, because there's not a ton because of the bug thing. But there were a few studies that said at the base of the tree, it should be around 500, which is, for reference, the same sugariness as a Coca-Cola. So what we found is it's 3 times sugarier than that in the leaf. This is big. Remember I said sugars and salts are great for holding on to water. So this is really great for the phloem. It means that the phloem has a very high sugar concentration, which gives the tree resilience in the case of drought. It can handle losing water because it has this amazing ability to draw it in through osmosis. And in another experiment, we found that this concentration actually can increase during drought. So not only is it high, it can get even higher to protect the plant. So we're working on this now more here at McLeish. Shout out, where's Paul? There he is, the great Paul Wetzel. So we're doing this at the tree. Thank you, Paul. We're making these small holes in his beloved trees, and I call this experiment "Be the Bug." This is our little nuance. Okay, so we made this is okay. Okay, stay focused, Jess. Okay, so Flora Meeker is the amazing Smithy who's doing this work down here for their honors thesis, and it would not be at all possible without the great Maxine Walden up here doing the high part. Yeah, shout out. And so what we do is we make these tiny little holes, and you can't really see it on the screen, but this part, you can see it a little bit, is really wet. And so we dig in until we get to the phloem, which is the really wet part, and then we use this microcapillary to absorb it. And then we bring it back to the lab and we analyze it. And this summer, we're going to be doing this on all different types of species and furthering Flora's amazing work. This hasn't been done in like 50 years, so we're really excited about the results. And then another project we've done was the great Scarlet Ruby graduated. Sad for me, very happy for them. They did a great honors project where they were also looking at how this sugar concentration can change over time in droughts, and they found that it's important and very dynamic. So that's about the phloem. I want to talk a little bit about photosynthetic cells in the next couple minutes. So like I said, more sugars and salts are typically good when you're inside of the plant. And in this case, we're thinking about wilting, and it gives you greater resilience against wilting. So the more you can photosynthesize and generate sugars, the better. And resilience is a key theme that comes up in a lot of our work. We're really trying to study how trees can be resilient against climate change. And so I'm going to share another art piece made by Chloe Gold called Recovery Conduit. This piece was a meditation on the resilience of trees. It is a piece of wood that they cut into this specific shape and hung various sentimental items of meaning into the different voids. And to quote the artist, "The trees remind me that recovery is a process. It is ongoing. It is always. It is a liminal state we exist in, always at tension with hardship and softness. It is a calculated dance, much the same as the trees who coordinate photosynthesis and xylogenesis in a careful balance. In all, the piece is a conversation between damage and regrowth, and enmeshing of these two forces into something beautiful and precious." So this brings us to this theme of resilience, which is really the heart of most of our questions. So another question we're thinking about is, how do northeastern tree leaves vary in their ability to be resilient against wilting? In order to study leaves, you gotta get a lot of leaves. So we go into the woods quite a lot, mainly with this guy Jack, who likes to take selfies. He's a great collaborator. It's like a brother, I love him. Anyway, so yeah, there's a lot of Smithies in different iterations over the years and other amazing lab members. This was us getting poured on this past summer. We got through it, we got the samples. Anyway, okay, so we collect a lot of leaves, we collect thousands of leaves, and we bring them back to the lab to study them. And we process thousands of them in our favorite tool called the osmometer. She's very temperamental, but we love her. And we have two of her. There's two. So what that looks like is we bring the leaves back, we shake them around in a centrifuge to get all the juice out of it. We pipette the juice. Look at Bella pipetting the juice, putting it in the osmometer, and then getting our sample. And so this took years. We have been processing these samples since I've been here. And what we do to get through it is we center joy and play. And so here are some amazing lab members, Maxine and Flora. Sometimes you have to wait 10 minutes between each sample. And so they're like, we should just bring our instruments. And I literally couldn't have been happier. And here's Bella working really hard on the osmometer sample. Also, shout out, Bella made this gorgeous bulb. Yeah, I love her. Both Bella and the plant. Okay, so we have some data. Here's some data for you. So, okay, this is our first pass at the data, which is really the question of which species are more or less drought resistant. And so these are the species we studied. You might recognize them. They're the dominant northeastern trees. And what you see on this axis is a metric of how many solutes are in the leaves. So you have less drought resilience or less solutes up here and more drought resistance or more solutes down here. So the ones in white are more drought resistant for their leaves. They're more robust against wilting. So white oaks, black and red oak, shagbark hickory, pignut hickory, ash. And then we have the birches and maple and beeches are a little bit less resilient in terms of their capacity to tolerate water stress. And so our big takeaway is that we have very large difference in species in terms of their leaf level drought tolerance, and we're working on scaling this work up right now. So this is the leaf level, and we really want to understand if leaf level manifests at whole tree level. So we're doing a lot of tree coring, which, yeah, got to stay tuned for that stuff. Okay, and so somebody who processed a lot of these samples is the great Al Torrens-Martin. Al has been working on a series of zines called In the Pines, which is about their relationship to pines, both personally and research-wise and musically. There's playlists in the zines, and I just want to read a quick excerpt from it. So this is from around here. Drought occurs when there is not enough water in the soil for a plant to take in and maintain its function, and its cells become shriveled and dry. The Turger loss point, or the amount of solutes, of a plant is a measurement of its tolerance to this dryness and whether or not its cells can bounce back from such an ordeal. Drought is also when you need something you cannot have. It is the lack of something that once made you whole. Drought is the loss of a family member, the sudden dark that comes over you when you realize you can never talk to him again. These zines are unbelievable. You can see just all the thought that goes into it. This is only a few of the pages. We have one of them down here, the newest edition. And you can see that they put a lot of personal touches on it. I'm obsessed with the playlists. So anyway, this is another product, amazing product that Al has made. So in conclusion, the current conclusions, we have the question of when water is being lost, how can the phloem hold on to it? And we found that there's just a really high sugar concentration, which was higher than what we were expecting. And we also found that it's dynamic and can change and increase to protect the plant against wilting. And this allows the phloem to hold on to water in stressful conditions. And when water is being lost in the photosynthetic cells, how can they hold on to it? We found that there's a big variation in their capacity to hold on to this water. We found that certain northeastern tree species contain more sugars and salts than other species, allowing them to be more robust. And so generally, for this presentation, we've been thinking a lot about what does it mean to hold on to something being lost. As a region, as a community, as a plant, as the phloem, and as a person. And through appreciating parallel forms of experiencing loss, we hope to expand our and your way of knowing plants and the planet. And so for further engagement, this is just some of the projects that we're working on in the lab. And so the amazing lab students and lab members have made different zines about some of the different projects. And so they're down here. Please just take one. There's not a ton of them. But we're also going to post them online so you can see them. But so just to show you what they are. So Chloe Gold made this gorgeous zine about how to love and lose a tree. Marge Poma Alarcon made embolisms and the optical vulnerability method, which is about their master's project, and xylem, which we didn't even get into. So this is cool. I mean, it's all cool. And Iz Thompson, a recent graduate, made a zine called The Place Lab Book. So Iz has really spearheaded our efforts to codify our lab practices. And so they made this gorgeous summary of it here. Bella Ivy made this great cloning trees zine. So this is a project Bella's spearheading in the lab, trying to figure out how to clone trees so then we can mess with them and understand what is genetic and what is not genetic in terms of their shifts due to climate change, adaptation, acclimation stuff. And Al Torrens-Martin did another amazing In the Pines, issue number 4, called Wintering, which is about the relationship between pines and winter. And our amazing postdoc, Malini, made a zine called Frozen, Thirsty, and Still Alive, a shout out to the conifers about her amazing work over the past year. We also there's an amazing art exhibit coming up by Avery Motz, who's in the lab. It's called Fieldworks, and it's an exhibition at the intersection of plant physiology, conservation ecology, and studio art, featuring botanical illustrations created with handmade inks sourced from sustainably harvested plants. And here are some of just a preview of it. And it will be on view in the Nolan Art Lounge from April 16th to the 30th. And last but not least, we don't take any credit for this, but I'm just so excited about it. There's going to be Art at the Bulb Show, which there has been for the past few years, spearheaded by the great Lily Caron. And the art is made by Jamie and Ella, and you can see some of it here, and you will see it in a second when you go over. All right, so with that, that's the end. Thank you all so much. Thank you, the Place Lab, of course, the Botanic Garden, the Bio Department, the Holbrook Lab, the NSF, and the USDA, and thank you for coming.
Thank you, Jess. We do have time for questions, but I just have to say I've never felt as much joy listening to a plant talk, and I've been to so many of them. Thank you for lifting us up, just like you lift up your team and everybody around us. So, we do have some time for questions. If folks do, like 5 minutes or so, if folks want to ask a couple questions. I want to prioritize if students have questions first. Please raise your hand, and I'm going to bring the mic to you because this is being recorded, and I want folks to be able to hear the question, and also anybody here to be able to hear it clearly as well. All right, Marty. Good, Marty.
Hi, Jess.
Hi, Marty.
Amazing talk. As you know, I work in rivers. And in the Mill River, I notice some years it's really low, and other years it's a raging torrent, right? So it seems like we go back and forth and back and forth. And in particular, the fall seems to be swinging a lot more. Like the data that I've looked at, just based on when I'm collecting data, happens to be like either June or early fall, and the fall in particular has more variability. Mm-hmm. So you've talked a little bit about drought specifically, but I'm curious about the variability and the swings back and forth and how resilient they are to that kind of
They
thing.
being?
The plants.
Oh, great. Okay. I was like, don't ask me about rivers, Marty. Okay. So yes, variability I feel like is the key prediction. So we're gonna have, as we all know, these horrible, horrible floods that have been happening every other year, kind of in opposition to this drought. And so that's a prediction of climate change, that there's going to be more vulnerability. I do, in terms of handling that and like how the trees will respond. I mean, the flooding that we've seen has not been like Paul, correct me if I'm wrong, has not been as manifested in the forests. Like the forests are just really wet, and that's usually kind of okay for the trees. Like of course you have trees like in other areas that aren't the forest, but that's less of what I know, so I don't want to speak to that too much. But from my experience, the plants that we showed that kind of have more solutes, so thinking about the oaks, they can really handle these fluctuations in a way that I think some things like maples will be struggling with. But that's just, and again, right, this is physiology. So it's like that's true at the cellular level and the organ level, but we're really trying to get the answers right now to see how it scales. And so once we have the tree ring data, that'll help us better understand like the whole tree response to this variability. Yeah, thanks, Martin.
Hi, that was amazing. I have a question about the solutes.
Yeah, great.
I was wondering if any other chemicals or pieces of things impacted the ability of trees to hold on to water, not just salts or sugars.
That's a really good question. Yeah, so we have like, is a really good one. I love that. So we can think about like plant capacity, and so sometimes like the structures of the cells themselves could potentially influence the ability of the plant to hold on to water, and so some species will have greater capacitance than other species. Angie, correct me if I'm wrong. I think that's right. And so I can imagine that would also factor into their ability to hold and retain water. It's a great question. Hi.
wanted to say that your talk was really incredible.
Thank you.
And I was really curious about how your lab incorporates potential invasive species of trees, or how that incorporates into your research in place.
Yeah, this is a great question. We don't touch them. It's a really good question. So we unfortunately, only have so many hands and minds. I think invasive species are so important, and again, I'll direct you to the great Paul Wetzel to think more about invasive species. But yeah, so we really just think about the native tree species, or the species that are primarily on this landscape that are not invasive. But again, thinking about invasive language and stuff too. But yeah, it's a really great question.
Hello. I wanted to ask a question about one of your slides. You were talking about future climate scenarios where we do fight rising emissions, or versus the ones where we don't fight rising emissions. I wondered if you could speak to any of the climate solutions that plant scientists are theorizing for the future.
Yeah, awesome question. So what are some solutions plant scientists are thinking about? Nature-based climate solutions. This is the big one. We actually just had an expert come and talk to our plant physiology class about it this past Thursday, so I'm ripe. Okay, but so what she's doing, she works at the Nature Conservancy, and they're looking at a lot of different ways to try and shift forest management practices to optimize carbon sequestration. And so they do a lot of really intense, robust studying of like where they can make the most difference. So they're like, if you're already crushing it in your forest management, they try and find places where they aren't, where they're just kind of clear cutting or doing something really intense to the landscape, and then they try through like financial incentives help people kind of shift their management strategies. And there was some crazy fact, plant physiologists correct me if I'm wrong, but it was like, I think if we optimize climate, nature-based climate solutions, it would help us help sequester 30% of the carbon needed to sequester by 2030. Thanks. Yeah, so it's a really powerful tool, and a lot of conservation organizations are thinking a lot about nature-based climate solutions. And this work kind of feeds into it and thinking about what are the species or individuals that can be most resilient, and so that'll help inform the forest management practices. Yeah, thanks.
Hi there. In your graphic where you did a comparison between the diffuse porous and the ring porous trees.
Yes. Yes, I didn't even get to talk about it. I think I planted you.
I'm not a plant. I'm a person. Was there a reason why you didn't look at sugar maple in the?
Oh, that's a really good question.
I see beech as the the highest diffuse porous tree, but yeah, is there a reason why you didn't look at sugar
that's
maple?
a really good question. The forest we were studying, this work was from Thompson Farm in southern New Hampshire around Durham, and there's just not a ton of sugar maples on that landscape, so that's one reason. The other reason is they're really hard to measure for water potential. We're working on ways of modifying the technique for doing that, so measuring the water stress. But they kind of, as soon as you put them in the pressure chamber, all this stuff comes out, and so it's really hard to get exactly what their water potential is. But again, I said, we're working on it. I think we figured out some ideas. But yeah, it was mainly that it wasn't on this specific landscape in the Northeast. But we're gonna be working on it, I think, this summer. So stay tuned. Yeah, thanks for that question.
Thank you. That was so awesome.
Thank you.
I'm an artist who loves to nerd out about plants, and I feel like so much of what I have heard about kind of the intersection of art and science is about like alternative ways of communicating to the like non-expert public. Mm-hmm. But what I'm hearing from you is that people in the lab practicing art is kind of a way of retaining your humanity and mm-hmm. emotions and not checking them at the door. Mm-hmm. And I'm just wondering how that play, like, have you seen results in people's resiliency or like the quality of the work or the commitment that people are able to sustain through that practice?
That's such a beautiful question. And it is something I think we think about a lot, I'd say in the lab and also in the classes. Like because in all my classes, they make art as like the main projects of the class. And I leave it really open to the students to if they want it to be more of like an illustrative, so kind of more thinking about the communication side, or a more abstract or personal piece. And so usually in class, we have pretty wide range of it. I guess I haven't done any studies on it. I really honestly I've been thinking about trying to apply for grants to think about like actually kind of studying it in the classroom and specifically in this context. So hopefully in the next 5 to 10 years, depending on funding, we'll do something like that. But personally, I know it's changed my life in being able to have the artistic component alongside the scientific component. I don't know if any of you want to speak to that, but you want to? There's a mic. Yeah, great. What's it, Chloe and Avery?
I mean, I also want to highlight something that is like in the lab is that 20% of our time gets to be invested in art. So, you know, like as a low-income student, I get paid to do art, and that's one of the most incredible things and does help me preserve my humanity at a place like Smith, which is amazing but very rigorous. Mm-hmm. And so that has always been like in your classes, the academic side and also this work side, a really big light for me. But also, yeah, I would say like my whole major has been really centered around this intersection of botany and the humanities. And just in hearing you say that, I'm thinking about the first time that word, thinking like, oh yeah, mm-hmm. The reason that those two things work together is because it lets me be a person. And I feel like I can always bring all of myself to the lab, however that looks. And so much of that is just the way that I process things, which is creatively. And so Jess has always made space for that, and it just feels like it's this whole new side of science that I've never seen before. And yeah, I guess this is turning into a love letter to lab, but like, yeah, I would say like it for like student testimony is the reason I think I've succeeded at Smith is that I've had that combination. So. I bet Avery's going to probably follow that up.
I second all of that. And as an example, I remember my first semester here in Jess's class, understanding climate change through plant biology and the arts. Yep.
It's long.
We were learning about, we got to the section about tree mortality, and it was so sad. And it was a whole week of learning about how like all the trees are dying. And it was a particularly hard week for me for some reason, like in general. And like I couldn't, it's hard to take in that kind of information. Like we see these figures and it's like, it's so difficult to comprehend. And the only way I could really process that emotionally was by making art. And so that has made me a better scientist because I'm able to internalize these facts and these figures and these realities and actually do this research, whereas like before it would just be paralyzing to think about these things. So it helps to kind of move into action and community.
Perfect note to pause on. Will you be coming down to Lyman?
Yeah, of course.
Awesome. So people can write more love letters to Jess down there. Please join me in thanking Jess one more time. That was a really, really amazing talk.